This protocol utilizes wearable optical probes to evaluate brain blood flow during the resting state. An important advantage of this technique is its portability, making it well suited for bedside monitoring.
The wearable probe allows resting state functional connectivity measurements to be obtained in the natural environments of the subjects for both diagnostic and therapeutic applications. This protocol utilizes wearable optical probes to evaluate brain blood flow during the resting state functional connectivity measurements.
Demonstrating the procedure with Chien Poon will be Ben Rinehart, a grad student from my laboratory.
At least 10 minutes before beginning the analysis, power up the FT-FNIRS and DCS with the light, modulation, and detector voltage. Use a tape measure to measure the distance between the nasion and the inion on the subject's head. Using the nasion as the starting point, use an ink marker to designate the location that is 10% of the distance to the inion. Place an EEG 1020 cap onto the subject's head, such that the marked point is between FP1 and FP2. Mark the point between FP1 and F7 on the left cortex, and the point between FP2 and F8 on the right cortex, to mark the boundaries between the superior prefrontal cortex and the dorsolateral prefrontal cortex, and between the dorsolateral prefrontal cortex and the inferior prefrontal cortex for the left and right hemispheres, respectively. Using a 3D printed probe, connect the multi-mode fibers to the 785 nanometer laser light source. Then place another single